So my name's Aaron Whitman. I'm a senior research associate here at the Gulf of Maine Research Institute, otherwise known as GMRI, and I'm also a master's student at the University of Maine School of Marine Sciences. So my research in the Maine eDNA program is I'm looking to compare eDNA to traditional survey methods. One survey method is active acoustics, which is a fancy fish finder, and then the other is looking and comparing eDNA or environmental DNA to a beach seine. And what a beach seine is, it's a net that's about 150 feet long. And what we do is we have a person on the boat, and they take one end of the net and they get off on the beach, and then the boat makes a U shape, and then we have another person get off the beach holding the other end, and between the two people they pull in the net, all the fish get funneled to the end, and we get an assessment of what the juvenile fish population is in coastal regions. So sea bass is the Casco Bay Aquatic System Survey, and what that means is it's a long-term survey that we're on the 10th year now, and we have 12 sites throughout Casco Bay that we survey with a beach seine, and we're looking at juvenile fish distribution. Also have a jigging component where we have 20 sites offshore on cod ledges, and we're looking for adult populations through that survey. And we have an acoustics survey that's also out on the cod ledges, and what that does is it gives us an abundance of different fish out by the cod ledges. The part that I'm focusing on with Maine eDNA is the beach seining, and 3 years ago we started implementing water samples into our survey design. And so at each site, we take a sample 100 meters out, 50 meters out, and then right at shore. And this is just 1 liter of water that we just scoop up with a Nalgene bottle. And then we bring it back to the lab. We filter it down onto a 0.7 micron filter. And then that's all the DNA that was captured in that one water sample that we then run for species that we're concerned about or interested in. In my case, for this project, I'm looking at Atlantic herring distributions. You can detect herring schools by their behavior using acoustics. So these are herring spawning schools. And so what we do is we go out at night to do active acoustics along a transect of known herring spawning stations. And we're looking for a tight school that is just right off the bottom. And there's really nothing else in this area that kind of does this at this time of year, at this time of day. And so we can use that unique behavior of the acoustic signature of spawning Atlantic herring to say that we know that that's an Atlantic herring school, and then we are using the eDNA to compare and see if those two survey designs kind of match up. eDNA can complement another survey design and pick up something that survey is lacking. So in the case for seines, or in say like a trawl survey offshore, it would pick up the species that the net's missing. So the biggest kind of problem that we had in this is kind of the nature of fieldwork, and we can't always count on our focal species being there at the time that we want them to be there. So while we were out doing our samples in 2020 and 2021, the herring fishery has actually kind of collapsed, and we did not see any spawning schools of herring on our acoustics to actually compare eDNA to. We still went out there and we took all the samples, and a very kind of good outcome of this is that we were still able to detect herring even in small amounts. So we knew that there were some very small herring schools, but nothing that we've historically seen. And it was really nice to see that we can still pick up eDNA signatures of herring in the open ocean with just a liter of water. So it just kind of shows the power of eDNA when we are using it on a species, even if they're not in the abundance that we had hoped they would have been. If you understand how you can use eDNA and you can understand how it operates in different environments, you can create a better project and maximize your results based on which technique of eDNA you want to use and how you're applying it in the environment that you're applying it to.